Boa tarde a todos. Bem-vindos de volta ao Dia do DNA. A gente está começando aqui a segunda parte, a parte de tarde. Eu espero que vocês tenham gostado da parte da manhã. E quem puder, se inscreva no nosso canal. Tá? Aqui eu tenho a honra de ter o professor Benoit Labonté. Benoit, I'm very happy to have you here. I'm honored as well. Benoit is an assistant professor and research chair in molecular neurobiology of mood disorders at the Department of Psychiatry and Neuroscience at the Faculty of Medicine in the Laval University. He's also co-director of the neurological aspects of mood disorder axis. Benoit, it's your turn.
All right, thank you, Betty. Hi, everyone. So as Betty mentioned, I'm Benoit Labonté. I'm a professor at the University Laval in Quebec City. It's a great pleasure to be here today. I want to thank also Betty and the organizing committee for having me. And today, I'm going to present some data that we have generated in the lab recently. Some data that combines, I think it's going to be an interesting presentation because basically this is some data that combines a lot of behavior with gene screening. So trying to understand the role of specific genes in the regulation of mood behaviors with animal models of stress. It includes a little bit of functional, morphological, and physiological data. So I think it's going to be a very interesting overview of what my research program does and how this strategy can help in understanding better the molecular neurobiology of mood disorders. So basically, as I just mentioned, my lab studies depression. So depression, as everyone knows, it's a major disorder affecting a lot of people in the world. Only in the United States, it's more or less 15 million people. It's obviously a leading cause of disability worldwide, associated with high levels of comorbidity, for instance, substance abuse in males, eating disorders in females. It's also a disease that is more or less well treated. Like we know that 80% of those treated for depression will show a certain level of improvement in their symptoms, but the problem is that 2/3 of the people suffering from depression do not actively seek or receive proper treatment, which again raises the importance of continuing to study this disease. And one thing that I think is important is to bring your attention to the fact that females are 2 to 3 times more likely to develop depression. And this is something that in my lab we're studying in great details. If we're talking about depression in terms of sex differences, as I just mentioned, there's the incidence where females are 2 to 3 times more likely to develop MDD or stress-related disorders than males. But there are also differences in symptomatology, where women compared to men have more severe scores of depression, will also associate with greater functional impairment or either atypical depressive symptoms and higher rate of comorbidity as well. There are some studies that describe differences in response to antidepressant treatment. So but this field is controversial, and there are a lot of contradicting results that have been published in this field. And of course, there are differences in the activation of stress-related brain regions and pathways, and this is what I'm going to be talking about today. Here I'm listing 2 images of the activity of the HP axis, which is affected differently in males and females suffering from depression. But beside of course, the brain regions involved in HP axis, there are also other brain regions, including the medial prefrontal cortex, that is one of the main hub region playing a key role in the pathophysiology of depression. And here I'm listing some examples like metabolic, morphometric changes, but also associated with molecular changes and functional changes that I'm going to briefly introduce in the next slides. So basically, one of the first pioneering studies in this field, very influential, was the paper published by Ellen Mayberg's group in 2005 in Neurons, in which they showed that there was a rich field of research showing elevated metabolic activity in the prefrontal cortex of treatment-resistant MDD patients. And this was associated very significantly with the expression of anhedonic symptoms, anhedonia in humans. Now, what they showed here is that basically the insertion of a stimulation cannula in the cingulate gyrus to do the deep brain stimulation of the prefrontal cortex could be used as a therapeutic options, basically normalizing all the changes that were associated with, or most of the changes that were associated with the expression of the disease into more normal conditions. And this was also associated with remission in these treatment-resistant patients. In terms of morphological evidence, there are also studies, postmortem studies mainly, that reported decreased cortical thickness or lower neuronal density and smaller size of neural bodies across the PFC. There are also brain imaging studies that reported thinner cortical gray matter and PFC subregions in patients with MDD. At the microscopy level, some postmortem studies reported loss of spine and dendritic arbor in the brain of MDD patients, especially in the prefrontal cortex and even in specific layers. And these interesting evidence in humans were also replicated with different mouse models of stress afterward. In terms of molecular evidence, what my lab has been doing in the last few years is basically characterizing the transcriptional changes affecting different brain regions in male and female with MDD. And this is an example of what we have published in 2017 and that we're also working on in another project in my lab. It's basically mapping changes in gene expression. So we have a lot of genes, for example, here in BA25, so the medial prefrontal cortex in humans, differentially expressed in males suffering from MDD, a lot of genes differentially expressed in females suffering from DDD, very low overlap between both sexes, as we can see here. When we create the transcriptional organization of gene networks, we can see similar components or similar functions affected in men and women with MDD, but through the activity of different genes. And this is basically has been shaping a lot the direction of my research program, because overall, at the end of the day, both males and females express more or less similar symptoms, more or less same functions are affected, but through the action of different genes. And so this is what we're studying, and this is also in support of how the prefrontal cortex seems to be affected in both males and females differently. In terms of behavioral and functional evidence, again, some work that we have published in Nature Medicines in 2017, we identified a female-specific hub gene. So in this case, we can talk about DUSP6, downregulated in the prefrontal cortex of women and with MDD and stressed female mice. When we did the downregulation through with a viral approach of DUSP6, we could see that we could promote the expression of anxiety and anhedonia in females, but not in males, as we can see here. And interestingly, this was associated with changes in functional activity of these cells. So combining these downregulation with electrophysiological recordings, we could see that basically the knockdown of DUSP6 by increases anxiety and anhedonia by changing the activity of pyramidal neurons in the prefrontal cortex in a sex-specific fashion. Suggesting that maybe, in fact, all these changes happening in the PFC may lead to a hyperactivity of this region. And this is, again, what I'm going to be talking about in the next few slides. In terms of functional evidence, just to support, again, this idea, is basically some groups, Immunifluency in 2016 published some work in Cell showing that the optogenetic activation of the prefrontal cortex decreases preference for sweet water in males without any change in overall consumption. Also decreased the same activation pathway, activation of the same pathway also decreased social interactions. And again, suggesting that if you are to increase the activity of the MPFC neural activity, this may promote the expression of depressive-like behaviors in males. Similar work also, but in this time, instead of activating the excitatory neurons or pyramidal neurons in the cortex, inhibiting the GABAergic neurons in the cortex, generated some very similar results. So this is some work that was published in the lab of Etienne Sibyl at this time when he was in Pittsburgh. Now he's in Toronto, and showing that basically acute pharmacogenetics or DREAD inhibition of somatostatin GABAergic interneurons induced anxiety-like behaviors in male mice. So basically reducing the time spent in the open arm of the elevated plus maze, increasing the latency to eat in the novelty suppressed feeding test, and reducing the time spent in the center zone of the open field. Basically, what this paper suggests is that by inhibiting the GABAergic system, there's a shift toward excitation in the prefrontal cortex, and this may lead to the expression of anxiety-like behaviors in male mice, again, with no idea basically what's going on in females. But as it is always the case, there are also other studies that are showing opposite results. For example, this case, Herb Covington in the Nestler lab in 2009 published some work showing that 10 days of chronic social defeat stress decreased social interactions and sucrose preference as expected. This is the model of social stress in mice. But the acute optogenetic stimulation of the MPFC neurons, basically in this region, rescued these effects. So basically, while from the beginning I'm talking about hyperactivity of the prefrontal cortex leading to depressive and anxiety-like behaviors, in this case, activation of this pathway basically generated an antidepressant or pro-resilient effect. So how is this working? Basically, the cortex is a very complex brain region as listed here, and this is some all of this is described in one of our recent review published in Frontiers. Basically, what we can tell with this is that the prefrontal cortex acts as a hub region involved in several fundamental cognitive processes, but also in the regulation of emotional processes. By integrating multiple sensory and emotional information throughout its input, so basically we can see input to the PFC from several other brain regions, integrating either a layer 2 or 3 and or 1 and even 5, and then being processed internally with interneurons and connection from other brain regions to be at the end output or sent out to control the emotional responses by modulating the outputs to other brain regions, as we can see here, including the nucleus accumbens or the ventral tegmental area. Well, this is like a complex slide, and of course, I'm not going to go into the details of all of these things that are listed here, but just to show that the importance of the MPFC has been put forward by several studies that targeted specific circuits, either going through the PFC or leaving the PFC toward other brain regions. And all of these are not clear yet as how stress affects the cells that are integrating information from outside the PFC in order to send it out once modulated appropriately to other brain regions. And in our lab, we're basically interested in studying the projections from the prefrontal cortex to the nucleus accumbens, and also the prefrontal cortex to the ventral tegmental area. Evidence to show that these pathways are important in control of stress comes from previous studies from Rosemary Bagot in the Nestler lab again, showing that increasing glutamate release via the acute optogenetic stimulation of NAC terminals, MPFC terminals in the NAC following defeat acted as an antidepressant effect. So basically, the social withdrawal induced by social defeat was rescued by this pattern of activation with no effect on forced swim tests or the open field or results. There's, on the other way, some findings that showed from Murugan, published in 2017, that showed that if this pathway is activated, prefrontal cortex projecting to the nucleus accumbens, there's a decrease in time spent with the social target, with no change in when the prefrontal cortex to the VTA was stimulated. In fact, the prefrontal cortex to VTA projection seems to be controlling more the speed at which the animals are discovering the environment and interacting with social targets. So this again suggests that activation of the corticoaccumbal pathway, but not the cortical tegmental pathway, may promote social withdrawal in males. Now, if I'm summarizing all of this, basically we can say that chronic stress in human and mouse is associated with morphometric, molecular, and functional alterations in the prefrontal cortex. There's evidence suggesting that hyperactivity of the MPFC may promote the expression of stress response. However, as I mentioned, there are contrasting results suggesting the opposite as well. Of course, the MPFC is heterogeneous with different subpopulation integrating input and projecting outside the brain regions. And there are evidence suggesting that cortically driven dysregulation of these subcortical circuits, including the accumbal and tegmental pathways, may be underlying the expression of depressive-like behaviors in humans, and also the expression of stress-induced depressive-like behaviors in animals. Now, the question that we have in mind here in the lab is how do chronic stress impact the morphofunctional and molecular properties of these two pathways in males, but also in females, and if these are affected the same ways. So to address this question, the results that I'm showing now have been published last year in Biological Psychiatry. Basically, we used a viral strategy, a transsectional viral strategy, to label these cells from the cortex projecting to the nucleus accumbens in red, and the same cells from the prefrontal cortex projecting to the VTA in green. And after being able to highlight these two cellular populations, we used a paradigm of chronic variable stress to stress the animals, both males and females, and we combined these approaches with electrophysiology, morphological analysis, quantification of gene expression, and also validated the contribution of these pathways to the behavior with chemogenetic approaches. So this is basically the results showing that our mouse model is working well in both males and females. And this model involves stressing the animals. So we're using C57 black 6 mice. On first day, we apply foot shock to the animals. And on the second day, it's tail suspension. And on the third day, it's tube restraint. And we apply these three stressors over a period of 21 days, after which both males and females express phenotypes of anxiety and behavioral despair and anhedonia-like behaviors, as we can see here. So males and females will take more time to start eating in the novelty suppressed feeding test. They will spend less time in the open arm of the elevated plus maze. They will spend less time grooming in the splash test and taking less time before becoming immobile in the force swim test. So measures of behavioral despair, here measures of anxiety, and they will also consume less sweet water, so have decreased preference for sucrose as a measure of anhedonia. And what we can say is that basically this paradigm of stress induces an emotional phenotype in both males and females, as we can see here. So knowing that we were working with a good animal model, we then decided to test our approach to highlight these the neuronal cells projecting to the NAC or to the VTA. And to do this, we used a transsectional viral approach combining conditional viruses expressing FLIPO or CRE in retrograde fashion. So basically injecting a CRE, a retrograde virus being captured by the terminals in the VTA, going back to the prefrontal cortex, where these cells would be expressing CRE will become green with the expression of this virus, the AAV dio-EGFP virus. Same strategy, but this time with the FLIPO conditional expression in the terminals in the nucleus accumbens, going back to the prefrontal cortex, and then becoming highlighted with the use of this F-dio M-cherry. So basically what we see here is that cells projecting to the nucleus accumbens are labeled in red, and those projecting to the VTA are labeled in green. We can see some very nice topological organization of these two projections, as we can see here with a proportion of cells showing a specificity of layer also. So as I mentioned before, the first thing we did was to combine this viral and behavioral strategy with the electrophysiology recording. So what we showed basically here is that chronic stress increases the input, the excitatory input onto these cells in NAC projecting neurons in females, but not in males. In fact, what we see both in females and in males for NAC projecting cells is that there's a decrease in the inhibitory tone that affects, again, males and females, nothing in the VTA projecting pathway. But in the VTA projecting pathway, we can see an increase in the excitatory input or the increase in the excitatory tone coming from outside the PFC. And if we merge this together, we can see that basically CDS increases the ratio of excitation over inhibition. So there's a shift in the excitatory inhibitory balance in the NAC and VTA projecting cells, more importantly in the nucleus accumbens projections, by altering excitatory and inhibitory input to these neuronal populations, affecting males and females differently. So this was interesting and kind of goes well with the old story that I was mentioning at the beginning, that stress basically increases excitability of this region, either by decreasing the inhibitory input on these cells, but also increasing the excitatory input into these cells. Then we also tested whether these changes were also associated with morphological changes. And this is basically the viral strategy that we used. It's very similar to what I've just described, but in this case, we decided to go with NAC projecting cells and VTA projecting cells in separate animals to avoid large overlap and maximize our quantification. And we can see also, we can label the spines here as listed. So if we're looking at the results from the NAC projecting prefrontal cortex cells, we can see that males are affected, but not very importantly. There's a small decrease in the arborization. It's more distal to the soma in stressed males, but we can see a stronger decrease in the arborization or the dendritic complexity of these cells in the females, and this is more proximal. In contrast, in VTA projecting cells, we can see that males are very strongly affected. There's almost twice as few arborization of these cells in males projecting to the VTA, and a smaller effect in females. What is interesting also is that in the NAC projecting pathway, we a increased density of dendritic spine in both males and females. So this could make sense with the hyperexcitability effect that we observed in our previous analysis, which suggests that stress may change the functional activity of these cells by decreasing the dendritic arborization of these neuronal populations in a sex and a pathway specific fashion. Now, of course, the next idea was to say, okay, what are the genes that are involved in mediating these effects? And to do this, we started with a targeted approach, but in the later sections of my talk, I'm going to talk more about the genome-wide assessment of these effects. But to start with, we use the same viral strategy as described before, and then we fact sorted both neuronal populations. So in red, we can see here a very nice separation from the green cells highlighted. So the nucleus accumbens cells, projecting cells, and the VTA projecting cells here. And we isolated these cells to perform qPCR. And here we selected a series of genes involved in cytoskeletal plasticity, for example, Omer, Shank, DGL4, actin beta, but also neural activity, so different subunits of NMDA receptors or AMPA receptors, so GRIN 1 to A, GABAergic receptors subunits as well. And again, it's hard here to interpret all of this in a short amount of time, but what I should say is that, again, CDS induces a pathway-specific transcriptional change in genes that are related to either cytoskeletal plasticity and neural activity that are affecting males and females differently. And this is true even when you'll see that we're using the same approach, but genome-wide. Now, so basically, we had at this point ideas that there were functional changes associated with morphological changes, also associated with gene expression changes, all of this affecting specific pathways in males and females differently. And the next question was, if we were to stimulate this pathway, could we confirm, in fact, the role of this pathway in mediating depressive-like behaviors and try to reproduce what stress does when we apply CDS for 21 days. And so what we did is we targeted our approach on the nucleus accumbens pathway because of the effects that were stronger in this case, and also because of the effects that were affecting males and females differently. And we used this approach the same approach, but this time to express a excitatory dread receptor in the cortex cells projecting to the neck. And what we can see here is that, application of the agonist in that strongly increased the activity of these cells, and when overexpressing the excitatory dread and when expressing the inhibitory dread, that application of C21 drastically inhibited the activity of these cells. So using this approach, knowing that our mouse model was working well, knowing that our viral approach was working well as also, we decided to start by stimulating this pathway. And what we did first is, and then this is not listed here, I'm just telling you, we started by expressing this channel, waiting for 3 weeks for it to be expressed at high levels, and then stimulating acutely, and we saw no effect. So acute activation of this pathway does not lead to any anxiety or depressive-like behaviors in males or in females. So then after this, what we did is that we overexpressed the same excitatory dread receptor or channel, and then combine this with a subchronic stress. So subchronic stress is basically the same type of stress, but it's administered for 3 days, which by itself does nothing. It does not induce 3 days of stress, has no effect on behavior. But the idea was that if we stimulate this pathway combined with 3 days of stress, can we increase susceptibility in order to see the expression of depressive-like behaviors? And this is basically what we've seen. So if you look at the vehicle, you can see no effect in the anxiety level, in the behavioral despair level, or in the sucrose preference. You can see both in males and in females, no effect. But when you stress the animals for 3 days and you combine this with the chemogenetic activation of this pathway, you can see that both males and females will start expressing anxiety-like behaviors and also some certain level of depressive-like behaviors as assessed in the splash test. So this activation of non-projecting PFC neural pathway combined with subthreshold induces anxiety and behavioral despair in both males and females. Now, the other way around, if we are to stress the animals for 21 days and then block the activity of this pathway, can we rescue the effect? And the answer is in male, no. As you can see, 21 days of stress with no stimulation induces an anxiety-like behaviors in the novelty suppressed feeding test, elevated plasmase, also forced swim test or splash test, so behavioral despair, and anhedonia. That is not rescued by the administration of C21 or the acute inhibition of this pathway. Now, very interestingly though, what we saw is that the inhibition of this pathway did rescue anxiety and depressive-like behaviors in females. As we can see here, administration of C21 or inhibition of this pathway did rescue anxiety in the novelty suppressed feeding test or the elevated plasmase. Also brought back to control level the grooming time and the splash test, with no effect on sucrose preference. So suggesting that the chemogenetic inhibition of this pathway rescues anxiety and depressive-like behaviors, but only in females, not in males. So this is basically a summary. I'm not going to go into the details of this slide, but this is the model that we are trying to put forward based on the results that I've just described. Basically, there's a reduction of inhibitory input, as we can see here, on NAC projecting MPFC neurons in both males and in females, but not enough to trigger abnormal stress response. So when it's combined with increase in the excitatory tones, as we can see in females, then their shift toward excitation that is induced by CDS becomes sufficient to trigger the expression of depressive-like behaviors in females. And this is basically what we think we may have reproduced with our chemogenetic activation. So basically, stress or activation of this pathway leads to the expression of anxiety-like behaviors in both males and females. But because there's only a change in inhibitory input only a change in the excitatory input in females, this is not when we block the activation of this pathway, we can rescue only these effects in females. And so this is what we think may be happening here as a model of why females are more susceptible to the effect of stress in regard to these two pathways. Now, the question is, after this, is basically, what are the true molecular mechanisms? Because yes, I showed some results here listed here in terms of specific genes affected that may be explaining these effects, at least in terms of physiology and morphology, but it is most likely involving much more complex transcriptional alterations. And this is basically in the second step of this project, we have decided to look forward. And these are data that are in preparation in the lab. So basically, to look at transcriptional regulation of these two pathways, we use a ribotag approach. So in the lab, we have a ribotag mouse. So it's RPL22 gene that is flux and has a HA tag. So when CRE is administered, the HA tag can be expressed under exon 4. And so basically every ribosomal subunit of this gene express HA tags. So they're bound to RNA. We can do an immunoprecipitation with an antibody against HA, and then purify the RNA that was actively expressed or translated at the moment of the sacrifice of these animals, and then perform RNA-seq. But basically, we use this approach to target NAC projecting MPFC cells and VTA projecting MPFC neurons after 21 days of stress. So sequencing control animals and stress animals, males and females, for both pathways. And here is basically the validation of our viral strategy. So here I'm showing results from the differential expression results or analysis. Basically, we can see in NAC projecting neurons to the PFC, we can see a lot of genes differentially expressed in males, stress males, a lot of genes differentially expressed in stress females, and a certain level of overlap, like more or less 20% of the female genes are shared with the male genes. And what we can see also are some functional terms enriched for these genes. And as I mentioned before, and as we showed in our previous analysis published in other like biological psychiatry or in Nature Medicine, we can see that very similar pathways are affected in males and in females, but again, through different genes. When we look at the genes affected in VTA projecting cortical cells, we can see a little bit fewer number of genes affected in stress males or in females. The overlap is also very smaller. And basically when we're using it, when we're looking at it in terms of a rank hypergeometric overlap analysis, we can see a total lack of signal. So this was interesting, suggesting that yes, in fact, not only this brain region as a whole is differently affected by stress in males and females, but also the transcriptional profiles in subpopulations projecting to different structure. Then this is a validation that we have done. So we use the same approach as described before. So the same viral strategy. We sorted out projecting cells to the nucleus accumbens and cells projecting to the VTA. And what we can see here is very nice level of validation for 6 genes that I'm showing here. So neuritin 1, neurogranin, reticulin 4, the receptor for the reticulin 4 gene, RAS GTP binding or RAS-related GTP binding protein B, neuroglin 3, the opposite strand. So this is a link RNA and a glycoprotein NMB that is coming out in males. And what we can see here is the first arrow is how it was predicted in the RNA-seq, and the second arrow is how it was validated in the qPCR analysis. What we see is that basically we did not see anything before at the RNA-seq level for GPNMB and VTA male stressed, but we did see an increase at the qPCR. Same thing for NRG3 opposite strand. So overall, if we're looking at this for these 3 genes, we have, for these 6 genes, we have about 92% validation, which is very, very good in this case. And then, of course, as this is something that my lab does a lot. So instead of focusing exclusively on changes in gene expression, so looking at conventional differential expression analysis, we move forward with a network analysis. So in this case, we're talking about weighted gene co-expression network analysis. And basically, it looks at groups of genes that are highly co-expressed and correlated with each other as functional units of gene expression. So these genes are now becoming a network, and they can be given a color. For example, as we can see here, and this is like the network structure for corticoaccumbal neurons. We can see the different networks that we have identified. Here, I'm listing each of these networks, whether or not they are associated with the effect of stress. So if we're looking at males network, we can see that 20% of these networks are affected with stress, by stress, and more or less 25% of the networks in females. And here I'm listing the Saddle-Brown network, which is the most strongly, is the network with the strongest association with stress in males. I'm going to talk about this in a few minutes as a very, very important and interesting network to explain the effect of stress that we have observed. And very quick, what we can see here is the same network structure in the cortical tegmental neurons. And interestingly, here we can see that only 5% of the networks are associated with the effect of stress in males, and 35 in females. So again, just to highlight how dimorphic the contribution of these pathways are and how stress will affect males and females differently. Here is basically a circles plot that highlights each part of a pie. For those that are not used or familiar with this type of graph, each part of the pie represents a network with a color here. And then, for example, this is the Saddle-Brown network. So we can see the color Saddle-Brown. And each layer represents a enrichment for genes differentially expressed. So we start with female downregulated, female upregulated, and the combination of both. So we can see that the Saddle-Brown here and even the green one here are not enriched for genes differentially expressed in females, but they are enriched for genes differentially expressed either down or up or both in males. So this is something interesting. We can see very sex-specific patterns. So take, for example, this one here, cortical tegmental neurons, the networks here are males, here they are females, and we can see this type of pattern that shows how dimorphic these effects are in males and females, and how each of these networks may be contributing on its own way to the combination or the global behavioral alterations induced by stress. So I'm talking here very quickly about one network, the Saddle-Brown network that I have mentioned before. It is involved, excuse me, it is involved in functional terms related to cell sex signaling. So this is a very interesting network to look at the activity of these cells. It has associated with stress in males. Its module IgA gene was very significantly associated with stress. And it is also very strongly enriched for genes differentially expressed in males, but not in females, as we can see here. In this network, this is the representation of the network. Excuse me. And then we have XLR4B. XLR4B is a gene that caught our attention, first of all, because it's a hub gene in this network, but also, and probably more importantly, because it is the gene that came out as the most significantly differentially expressed in our list. XLR4B is an imprinting gene, so it's paternally repressed following dynamic and tissue-specific patterns. It has some homology with the human FAM9 gene family. And what is interesting is when you go on PubMed for this gene, you have only 3 papers. And one of it is suggesting that this gene may be involved in the control of dendrite and spine formation, but also regulating working memory deficits in mice and maybe being associated with autism and schizophrenia in humans. So in this case, we found it as being strongly overexpressed in cortical accumbal neurons, but also in tegmental neurons, in males only, and also, as I mentioned, as a highly connected hub. So it's a gene that is connected with more members of its network, and because of that, is likely to have a stronger impact on behavior. This is basically what we've done. Just going to go real fast on this to complete the presentation. Is we just tested if we were to overexpress this gene in males and females, both pathways, could we induce the expression of depressive-like behaviors? And this is, again, just to show like we have a virus that can overexpress this gene. When we overexpress it and combine this with no stress, just overexpression, we have 0 effect in males or in females. When we combine the same overexpression with subchronic stress, so 3 days or 6 days of stress in males, what we can see is the beginning of expression of anxiety in the novelty suppressed feeding test, and also in the elevated plus MACE test, some behavioral despair measured with the splash test or with the force wind test, and no change in anhedonia. And this is even strengthened after 6 days of stress, as we can see here. Interestingly, absolutely no effect in females. So this seems to be really driving anxiety-like behaviors in males when overexpressed in the NAC. Now, the same thing in VTA projecting cells. We saw absolutely no effect when expressed acutely with no stress combination. But when we overexpress it with 3 days or 6 days of stress, we saw nothing at the anxiety or behavioral level. But this time we saw an effect at the anhedonia. So basically, this pathway may be driving the expression of depressive or of anhedonic behavior in males and not in females. So this is something that was super interesting to us because it kind of confirms basically what we have observed in our mouse model after RNA-seq, suggesting that the overexpression of this gene may be driving stress susceptibility, but driving different components of a complex depressive-like behaviors. And here is basically the summary of what I've been showing. I'm going to skip this slide just for a matter of time. Okay, and just go to the overall message of my talk today. Basically, stress affects these 2 pathways, NAC and VTA pathways differently. It induces transcriptional changes, as I said, not only differential expression, but also changes in the transcriptional organization of gene networks. These changes are likely to be involved in the functional effects that we have seen, changing the excitatory to inhibitory balance in a sex-specific fashion, affecting more importantly the NAC projecting cells than the tegmental or VTA projecting cells, and leading ultimately to the expression of depressive-like behaviors. What we've shown is that the cortico-accumbal pathway may be driving, more importantly, the expression of depressive and behavioral despair-like behaviors, potentially through this gene, XLR4B in males. Also, I didn't talk about it, but NRN1, which is another target that we have looked at in males and in females. And also XLR4B driving anhedonia, this time when overexpressed in corticotegmental pathways. So this is overall the story that I wanted to tell you today. It really highlights the contribution that both of these pathways may have in driving specific aspects of the complex depressive-like behaviors in males and females, and also points us toward what type of strategy we should be looking at when trying to decrease the expression of these symptoms in humans suffering from depressive-like behaviors. And of course, I'm going to conclude here by acknowledging the contribution of my lab. This is part of my team. Most of the work that I've been presenting today has been done by Thibaut Bittard, a PhD student in my lab, and also Samane Mansouri, also in my lab, working more at the bioinformatic level, and also all of the funding agencies that have been supporting this work. And would like to thank you again for inviting me, for your attention, and I would be happy to answer a couple of questions. Thank you.
Thank you, Benoit. It was a very interesting talk. I have some questions. I don't know if we'll have time to figure them out. And it's very important to highlight that your research has going towards a direction that not nobody, but few researchers go through, that it's the sex-specific differences between men and women, like males and females. So it's very, very interesting. I one of my question is that so you saw the differences between males and females, but when you analyze the females, did you check about the menstrual cycle, if they were at the same phase of the cycle, so we can see if the difference you're seeing is specific of a cycle, of a specific phase of the cycle, or if it gets more heterogeneity in your sample. Do you understand my question?
Yeah, I totally understand. It's a very good point. It's actually a very important point also. So the answer is yes, we look at this. I did not mention it here, but every time we do some behavioral assays with our animals, we always smear the females so that we can evaluate at which stage of their estrous cycle they are. And in this case, we did control for that. Basically, there was no heterogeneity. All females were the same cycle, which raises another question: is if you were to look at them into another cycle, would it be different? Very good question again. And then there are other groups that have been looking at this, including the group of Marianne Senet, for example, and others. And the answer is it's hard to say, okay, but I would tend to say yes. There are differences in gene expression according to the estrous cycle. It's just that how different they are at the genome-wide level is still unknown, and how these changes can contribute to the phenotype is also not very well known. In humans, I did not talk about this too much today, but in humans, it's a little bit more complicated, especially when we're looking at postmortem samples, because we cannot really account. So what we can do is only with age, which may be representing more like a pre-postmenopausal status, for example.
So one question that was raised here is, are there differences found between men and women related to sex hormones? Was this considered in the in vivo study?
Yeah, again, this is a question related to what we've just said. So we always also look at baseline gene expressions. The models that we're using is also allows us to account for variation in baseline gene expression. So just genes that are expressed differently in males and females controls. So we can control for that, and we do control for that. So the findings that I'm listing, so for example, the XLR4B, okay, that I mentioned, is not expressed normally in males and in females. It's not expressed, zero. But when the animals are stressed, only males start to express it at high levels. And we can see like 0 counts versus 200, 300 counts. It's really a strong induction. I've honestly never seen this before. So it's kind of something that shows that it's not only related to stress, to hormone. It's also related to the effect of stress. But we're doing our best to account for this and not have a real. I wish I could have like these sequencing results for every stages of but no, I don't have that. Sorry.
No, it's good. It's good. And I have one more question. So did you try looking at the effects of antidepressant drugs? So do males and females respond differently after stress to antidepressant drugs?
So not in this case, and we have not yet done that in the lab, but this is definitely a direction that we are interested in taking. The idea really was to say, okay, what's happening in baseline when the animals are stressed? What are the changes in gene expression, epigenetic, etc.? And then let's see how conventional antidepressant strategies can help in rescuing the effects. Like I mentioned in my introduction, there are studies that suggest that male and female will respond differently to, for example, SSRI or other types of like conventional antidepressant therapies. But it's very controversial, and most of these studies are not powered enough to draw some specific conclusion. It's something that we will investigate more details.
Yeah, because you looked at differences in the transcriptome. So does it translate in differences when they are treated? Do they rescue at the same differences or not? Right?
Yeah, exactly. And say also what we've shown before, not here, but say resilience to stress, for example. When we're looking at it, it's important to understand that it's not only, we're not seeing the opposite. It's resilience is something completely on its own. And it's like the activation of specific programs that allow the animal to deal with stress. And so sometimes people have the idea that, oh, I'm just going to rescue what we see in susceptible animals. I don't think it's what we should be doing, in fact, is acting on the networks or the gene programs that are responsible for the expression of resilience and promote these for like an active coping strategy for stress, for example. But it's easy to say, it's difficult to do.
I have one more question here. The gene XLR4B looks pretty interesting. Do you think this gene could be associated with other psychiatric disorders like drug addiction when the subject has alcohol withdrawn?
Yeah, so I totally agree that XLR4B gene is very interesting. Just because look, the phenotype is so strong, and it, like I said at the beginning, it's a gene that is induced by stress, and then normally it's not. in this pathway, mainly. And this gene has been associated, so variation or SNPs in these genes have been associated with autism, for example, or with schizophrenia also. So I think it's totally reasonable to think that it may be associated with other psychiatric conditions, because more and more the genetic studies are showing convergence, or at least common grounds throughout different psychiatric conditions. So there's a core of genes affected, and depending on the environmental features that everyone is facing, may be developing into schizophrenia, depression, or, sharing common ontologies and alterations. And because of its role potentially in regulating spine morphology, maybe also regulating neuronal activity, and through like chromatin remodeling, it's not clear very well, but we're working on it, of course. I think it's definitely a possibility to see this gene being involved in other psychiatric conditions. Yeah.
Okay, so I have no more questions. It was a really interesting talk, and I'm very honored to have you here. I'm glad to see you again, right? And if anyone has one more question, no? So thank you, Dr. Labonté, and see you.
I see Carolina also. Hi, Carolina. So yeah, thank you all for having me. It was a great pleasure. Very happy. If you guys have questions, feel free to email me. If there are any students that are interested in coming to study, we have open positions also in the lab. So feel free to email me, send me your CV, and we can talk. It would be a pleasure.
Okay, thank you for coming, and see you later. Bye.
Bye-bye.
Pessoal, quem quiser, o próximo vídeo vai começar em 2 minutos. Obrigada.